a lot of the work I do is in the coevolution of geosphere and biosphere. I went to DALL-E, you know DALL-E, AI, ChatGTP, yes, you all know it. I went there and I told him, please, can you draw for me the elephant and the blind scientist? You know the story of the elephant and the blind man, how many? Nobody? Man, I'm getting old. So the story of the elephant and the blind man is that somebody got a group of blind men into a room with an elephant and asked them to touch and describe what they had in front of them. And somebody was touching the leg of the elephant and said, well, it's a tree. Somebody went on to touch the tail of the elephant and said, well, feels like a giant donkey. Somebody touched the how you call it in English? The trunk, and said, must be a giant snake. And everybody was describing the elephant in a different way. Why? Because they had just the perception of their little corner of what the elephant was as a whole. So what we do as a scientist, because we are trained in one discipline, we typically approach science from one side. And so I asked DALL-E to design the elephant and the blind scientist this time, not the blind man, because that's exactly how we are approaching science and Earth system science especially, from our own perspective. If you look for I was discussing with some of you before, it's a shame that it's great that we did reductionism and we made discipline of science over time, because the amount of information we need to learn for every discipline is increasing a lot. So we need to become an expert on something, but at the same time, we're not aware anymore of what the other sciences are doing. And a long time ago, we didn't have sciences, we had science, and science was actually philosophy. It was called natural philosophy. And we just spent 250 years breaking down natural philosophy into disciplines. And I think now it's the time we need to bring together the discipline again and reapproach things at the system level. And to a certain extent, that's what astrobiology is trying to do, right? You get to astrobiology as an expert in one discipline, but you approach it in a way that allows you to interact and integrate with the other people so that you see the entire elephant, not just a piece of it. And this type of argument, I think, should be more often discussed inside university programs and schools. So now that we've seen the blind scientist and the elephants, you should all have seen, now you know the story, right? How many know the story of the blind man and the elephant? Everybody, fantastic, love it. Now, let's do this. Have you ever seen this? How many of you? So painful to see so few hands up, and especially from people that probably have a bit more experience in this. You really never seen this. Many of you never seen this graph here. So it's something that is increasing, right? You all agree? This is probably the single most important graph ever made in the last 60 or 70 years. If you want a tattoo, you should tattoo this one. I'm not kidding. This is, some people tattoo the tree of Darwin, the sketch of the tree of Darwin. Some people have their own favorite molecule. This is the reason we are in trouble right now. This is what's happening right now. This is CO2 concentration over time at the Manual Law Observatory. This is a continuous record of CO2 concentration in the atmosphere, started somewhere in 1953 or 54, I don't remember exactly the date right now, by probably one of the most influential environmental scientists ever, which is Charles David Neeling. This is the Neeling curve. That's the name of this curve. So this person here started measuring CO2 in manual law and realized that yes, there was variation, but things were increasing. And now he's dead, but other people are keeping up his work, and we're still measuring it today. And you can go to the website of the Neeling curve, which is managed by NOAA right now, I think, or Scripps actually in San Diego, or you can go to Twitter and read exactly what's the concentration right now. In his lifetime, he was challenged many times because people told him, oh, you're measuring CO2 from the volcano, you are measuring CO2 from passing ships, you are measuring CO2 from fires, you are measuring other things. This is not atmospheric. And so what he had to do, he had to go to Antarctica, he had to go to South America, he had to go to a number of other places and repeat measurement almost daily for months to year in every location to show that this was not a local effect in Hawaii, but this is in fact a global effect. This is the rise of CO2 that we now link to climate change, global warming, and the ongoing climate crisis. Now, this is a single year right now. you see there's a certain periodicity here. And this is, you see, daily average, weekly average, monthly average. And you see how things go. And these are 2 years in a row. You start to see seasonality, right? Winter, summer, winter, summer. But things already show a trend here, just in 2 years. And if you put the entire record together, you can still see this jigsaw going up and down, saw-like pattern, but things have been increasing pretty dramatically. And we know this because we are measuring in the atmosphere continuously. If you take this, I keep saying this sentence every year, I'm realizing it might not be true anymore. I'm relatively young, and I might be close in age to many of you, more than you may think. And I can do an exercise. There's an interesting website where you say, you can do this. You do my life in CO2. I was born at 341 ppm, graduated 384, PhD at 400, researcher, assistant professor at 409, professor at 413, 20.8% increase in my lifetime. Repeat, I keep saying relatively young, yeah, I can still say that for a few years, but I had a 20% increase of the CO2 in the atmosphere during my lifetime. This is a huge increase. You can try your own increase up to where you are right now. I believe today we are at 456 something. Now I haven't updated these slides. This was November a few years ago. But we know this. This is the previous part I showed you, because direct measurements. And we know this because we can measure CO2 in a number of other locations. We can measure CO2 in the tiny bubbles trapped in ice in Antarctica and in glaciers. We can do it through trees. We can do it through other proxies. And you can see that, yes, CO2 has been increasing from Industrial Revolution going up, and then dramatically spiking. And we can go even further back. This is the last 1,000 years. Look how much variation there has been. When climate negationists and people that are against climate change, they say that doesn't exist. They say there's always been variation. It's very true. It's a matter of how rapid it is and how much it is. You can see the previous variation. Can you see the today line here? No, I can help you with that. Here where we are. You may have lost it in this graph because it's just a tiny dot, but look where it skyrockets up here. And in this debate, there are a number of other evidence that are very important and that I don't know many of you ever heard. So you all heard that CO2 is increasing in the atmosphere, right? Besides me telling you now, you heard it before, right? Everybody heard CO2 is increasing. How many of you heard that the isotopic signature of CO2 on the atmosphere has been changing? Okay, there's few people here. Perhaps the biggest evidence we have to say that the CO2 in the atmosphere is from combustion of oil and gas by human and not by volcano or natural sources is that if you go measure the CO2 in volcanoes globally as a very specific isotopic value, delta 13 to delta 13 carbon to 12 carbon ratio of -5. Mantle CO2 is pretty much constant globally. You go at the mouth of a volcano, you measure it, it's always -5. But we know because gas and oil are biological in origin, and we know life is very lazy in using isotopes and tends to make things lighter. If you measure plants out here right now, they're probably anywhere from -25 to -35 in ratio between 13 and 12. If you take petroleum as the same signature, it's very light, we say, because it's derived from old plant and old organic carbon that has been cooked over time. Now, if it's true, the majority of the CO2 coming to the atmosphere is coming from burning things that are isotopically light, the signature in the atmosphere should not be constant, should not be equal to -5, which is the mantle, should be getting lighter. And this is exactly what is happening. It's getting lighter and lighter and lighter and lighter and lighter. And you can do mixing trend. Say how much CO2 has increased over time. If this CO2 comes all from petroleum, how much lighter should we be? And they match perfectly. Now I realize this is a bit more nuanced than what we say to the public usually. We just tell them CO2 is increasing, is petroleum. Well, that's not the real evidence that is petroleum. We have much stronger evidence. We just don't approach it to explain it. And the beautiful things about science is that you might believe it or not, but it stays true. Don't believe to gravity and try to jump from a window. You cannot escape that. And all all this, as you know, has huge implications with temperature, and where we're going, and our projection of things, and so on. So it's clear that as humans, we are impacting our planet. I think there's no doubt about that. The facts are all well lined up. We have multiple lines of evidence. We can also say that we are impacting in ways that are different from the rest of life. We can also say that we are accelerating some of the rates that were not present before. It would be a mistake, though, to think that we are the only species that has an impact on the planet. That's something we hear sometimes. Humans are the only species that are destroying, ruining, changing, altering the planet. That's absolutely not true. Because every single life form, from the onset of life to today, has impacted and changed the planet. Now, everything I will say right now, you can take it two ways. You can interpret it as me providing evidence to climate negationists to say, oh, you see, humans are just doing what life does. Nothing to worry about. I'm not talking about that. We are doing things right now that are really problematic. And we are doing things right now that they're not problematic for the planet. They're problematic for our own survival in the next 100, 200, 500 years. So I never understood why we are having a question of us saving the planet, because it's not about saving the planet. We can wipe away every eukaryote on this planet. Life on this planet will keep going, microbial. It's a question of saving ourselves. So don't misinterpret me thinking I'm supporting climate negationists or of one way or another. I'm just stating true fact about how this planet changed over time. We are doing a mess. We are not the only one. People have done a mess. People. I keep saying microbes as people. Sorry. It's a relationship bias I have with them. Microbes and life had an impact on the planet since life emerged on this planet. Always. So if we are changing our planet, and all life on this planet always changed the planet, what can we learn from the past? There's anything good we can learn about how life interacted with the planet that might tell us not just something about where we come from, but about what we're doing, what we should be doing, and how to search for life somewhere else. Because in the end, as the question I got before is, okay, what can I measure that tells me that life is there? Because I'm not going to be able to measure life for a long time. So I need to find an alternative way of doing it. There are a couple of very important concepts in biology that are not really taught, not even in biology completely, just in certain branches. For example, in ecology, they're very well known, that I think everybody should know. Every time we speak about inheritance, right, what you inherit from your ancestor and from the past, the majority of people think about this. How many of you know what this is? Karyotypes. Everybody knows. DNA, these are our chromosomes, right? We say, I inherited my genes from my ancestors, and we pass genes on to future generations. In reality, inheritance is not just about genes. It's about a lot more than genes. Two concepts. First one, niche construction. It's a technical term used in ecology to say the life form can alter the environment to better suit their needs. We do it. We make roads, we plant fields, we purify water, we concentrate things, we extract things, but we're not the only one. Termites, super easy one. They for sure change their environment, right? They change the amount of oxygen that gets into the soil, they change the eating pattern, they change the gases, they change the landscape. They're not the only one. Beavers, they can change completely the way rivers go and where they go and what not. Oxygenic photosynthesis for sure changed the planet. Both biologists and geologists recognize it in my previous interviews, right? But you can do this thinking about how much an organism can change its environment, and you realize that every single organism, to a different degree, has an impact on the environment, and it's going to change the environment. So niche construction, it's a way in which you change and modify your environment and transform natural selection pressure in your favor. The other one is something called ecological inheritance. You don't just inherit the genes from your previous generation and the past and all the organisms before you. You also inherit an heavily modified environment. Why? Because you have niche construction. So our future generation, our children, will inherit a planet on the way we leave it to them. We know this, we say this all the time, leave a better planet to your children than you received it. But the thing is more intricate into life itself. Did you ever think about oxygen? I mean, you are all breathing oxygen right now. And you are sure you inherited genes to breathe oxygen from your parents, grandparents, from the evolution, all the way back to the emergence of aerobic respiration. But we did not just inherit a gene to breathe oxygen, we also inherited a planet where oxygen is now 21%. And this was not the way the planet was before. So we inherited the genes and we inherited a heavy modified environment. Ecological inheritance, not just genetic inheritance. Now, if you start to see the way life can modify the environment, and life passes on a modified environment and the gene necessary to interact with this environment, suddenly you are not just thinking about a planet that is changing in a cosmic trajectory dictated by physics, chemistry, and geology, and life that is running on top to scrambling and adapt to whatever the conditions are thrown in life. My life, a planet, they start to appear to interact, because life can change the direction of the planet. You know our cosmic temperature, our average temperature dictated by the distance from the sun. Yes?
Minus 15?
Yes, about minus 15 degrees. And yet we are not a frozen popsicle. Why? Why we're not? Because there are gases and there's a greenhouse effect, which is not necessarily always a negative thing. And this greenhouse effect doesn't come only by geology and volcanoes. There's also methane, which is a very powerful greenhouse gas, which is, for the majority, right now, and has been for a long time, biologically produced. And our average temperature is, in fact, more or less like plus 15, rather than minus 15. So we really impacted the planet on temperature-wise in many ways over the existence of life. And so coevolution is more something like this. You have life, you have environment, and of course life wants to have a high fitness on its current environment, is well adapted to whatever condition, but over time, one smooths out the other, and so you have organismal evolution and environmental evolution, and you have genetic you have genetic inheritance and environmental inheritance until one influences the other, until they keep staying, generally speaking, well adapted, well interacting to each other. Now, this simple idea of coevolution, one changes the other, you smooth up the surface where you are moving, and at the same time you adapt to the new angles you're creating, if you want to visualize it in some way, is at the core of what we call coevolution. Now, coevolution is a concept that we studied very, very well between organisms. Coevolution of colibri with their flower, coevolution of predator and prey, of pathogens, of viruses with the cell. There are many examples of coevolution. Has been only relatively recently that we are studying coevolution of life with the planet in a more systematic way. So if biology has influenced planetary evolution through time, how much? I think that's one of the big questions out there right now. How much has life influenced our planet? If we had to rethink this planet without life, where would it be as a planet today if there was no life present on it? Well, probably frozen, probably no oxygen. These are the easy ones. There's a number of other things that are not so well understood, and it might surprise you depending on your background. So let's go back to think about the biggest example of coevolution we have, which is the Great Oxidation Event, right? The evolution of oxygenic photosynthesis, because it's just one type of other photosynthesis. For the non-biologist here, we have many types of anoxygenic photosynthesis, many types of phototrophy overall, and then we have the oxygenic one that everybody's chasing today, but it's just the most abundant right now. It's not a given that was the most important. For sure, had a huge impact on our planet, 21% oxygen. But if I ask you, why do we have 21% oxygen on our atmosphere? How many of you will say because we have oxygenic photosynthesis? I know it seems circular as an argument for now, but try to think. If I tell you why we have oxygen on the planet, how many of you will reply because we have oxygenic photosynthesis? Okay, not everybody. I will be curious later to know why you're not convinced. But we know it had a huge effect. We know we went from very reduced to oxidized. We know we had probably 1 or 2 or more bumps over time. We know these influences a lot of things, right? From just being reduced to oxidized to a lot of other things. But you know this equation, textbook, right? Oxygenic photosynthesis, CO2, water, light, sugar plus oxygen. This is what most people study on the why we have oxygen on this planet. But you also should have seen this reaction here. This is heterotrophy, right? Sugar plus oxygen equals CO2 plus water, and it provides you energy. If you look at them, they're actually the same identical bulk reaction, just run in reverse. Which means, theoretically speaking, that photosynthesis and respiration are doing one the other. They're doing the same things, but in different direction. They're one the other face of the medal. And the problem is that if every oxygen you're making is respired somewhere, how much oxygen we should have in the atmosphere? 0. So the accumulation of oxygen in the atmosphere is not an effect of biology, not alone of biology at least. It has to be the interaction with something else. And what's the easiest way to increase oxygen over time? Well, one of my mentors, Paul Falkowski, would say, you hide the candies, you hide the sugar, you take it away somewhere. And where can you take it? Well, you can put it in cellulose and plants. You can put it underground, bury it deep underground. You can recycle it deep into the crust. You can push it into the mantle through subduction. One way or another, if all the organic carbon photosynthesis does is available for microbes to respire, you're never going to get any oxygen, right? So the accumulation of oxygen itself on this planet is a geobiological effect. You need oxygen to be made, and at the same time, you need to remove the counter reaction one way or another. You need to slow it down. You need to change it in a way that allows you to accumulate oxygen. So biosphere, through photosynthesis, oxygenic, and geosphere, through organic matter burial and subduction, have contributed to the net accumulation of oxygen in the atmosphere, decoupling photosynthesis and respiration over time. Otherwise, photosynthesis alone doesn't explain 21% of oxygen on this planet, right? Now, this is super nice because before, great oxidation event was linked to the emergence of oxygenic photosynthesis. Invention, evolutionary invention, I mean, and we get oxygen on the planet. Well, no, you cannot have it unless you hide the sugar somewhere, and now you can have it. It's a geobiofeedback, so it looks already more intricate, but it's even more because as soon as you start to pump and increase this up, well, oxygen is a nasty compound, can make a lot of reactive species, can destroy protein, can destroy DNA, can destroy lipids. Now, we all know we are well adapted to fight against the reactive oxygen species, radicals. We even sell creams and things to help you fight against radical and age less. But we know that these and all the enzymes we use to do this, and life uses, are a late adaptation. Can you imagine the first time oxygen started to accumulate on this planet? People call it great oxidation event. Some of my colleagues call it the first great extinction event. All the organisms that were not able to cope with the oxygen, they either died or they had to escape somewhere, like deep sea hydrothermal vents, subsurface, anoxic environment. Possibly the first mass extinction of microbial life on this planet, not witnessed, not discussed. If you take a paleontological book about the great extinction, it's not mentioned anywhere as a great extinction because they're focused on eukaryotic life. But that's one of the points where probably we lost the majority of the complexity and diversity we had accumulated at the time in terms of life. So we have oxygen, we have the planet interacting with it, and then we have the first mass extinction. And going to reduce to oxidized means also suddenly you are changing the relative portion of electron donor and acceptor, getting more and more oxidized things over time, which means that you can do different reaction. Because before you can do maybe hydrogen to CO2 to make methanogenesis, because you are in this way. This is a small jump in the electron tower, so not a lot of energy. But as time passes and you get more oxidized compound, you're starting to do bigger jump in what we teach in undergraduate textbook, which is the redox tower, where you can couple metabolism, or any redox couple for that matter to calculate delta G, and we get more and more metabolism going. So we're oxidizing the surface of the planet. I told you we're hiding the sugar in the crust, in the mantle, which means we are subducting reduced carbon, which, if you're a geologist, you know has huge implication for composition of the magma, redox state of the magma, type of volcanoes you can get, type of volcanic activity. There's a huge change in what you're recycling to the surface. You are changing what redox couple you can have to biology. You're changing the minerals. How many of you heard about mineral evolution? Well, not enough. There has been work done to show that 50% of the mineral on this planet right now, they're directly linked to biology. If there was no biology, you would have never had them. And of these, another 30% happens only just, no, not 30%, a lot more, sorry, from 1,500 to 4,000 mineral happens just thanks to this change in redox of GOE. So if you go on our early Earth, calculations show that perhaps you have 1,200 to 1,500 different minerals, no more. It's not a very mineralogically diverse planet, considering that now we have over 5,000 or close to 5,000 different minerals. So GOE didn't just change what redox couple biology can use, didn't just change the fact that we had oxygen, because oxygen means sulfate, nitrate, nitrous oxide, and so on. Also changed what gets subducted, which changes and influences plate tectonic. It changed also what minerals are possible on the planet. With all the implication we discussed before for cofactors and evolution, interaction with your redox couple, organometallic catalysis and mineral catalysis. So there are big feedback going around. For example, pyrite, which is very little abundant, which is the most abundant sulfur and iron mineral at higher oxygen concentration, was probably less abundant before when uraninite was present and when siderite was present. Siderite is very stable at low oxygen concentration, almost zero. But you just go above 0.001% of present atmospheric level of oxygen, and siderite is unstable, doesn't exist anymore. So we had an evolution of mineral over this time. We had an evolution of the metal that could be used. These are the same protein, some of the protein I was discussing earlier. And as you can see, some use iron sulfur, some use copper. And they need to use it because this one is interacting with nitrous oxide, this one is interacting with sulfate. So they need different midpoint potential to exchange the electron. Well, if you go back to the redox tower that is in every textbook, and you think about interaction between different redox couple, depending on the redox you want to use, you need a different metal. Some metal are very good for some couples, but they're terrible for others. And you can see that some act on the very reduced compound that were abundant early on this planet, but some are needed to access the more oxidized one, like copper. You all are using copper right now in your body to respire oxygen. Your terminal protein respiring oxygen is a copper protein right now. The interesting thing is that you don't just need to have an exact metal to do a certain job. This metal changed over time a lot. For example, iron was very abundant early on in during the history of this planet. The ocean were filled with iron everywhere. And then after GOE, iron went from 10 to the minus 5 all the way to 10 to the minus 9 in the ocean. Huge drop in concentration. While other compounds, so you see specific molybdenum and copper, for example, that are needed for some things, really went up. Look at this, from minus 20, 22 to minus 7. That's order of magnitude increase in the abundance. This is because of their chemical properties, the way they interact in minerals, their solubility due to the redox state, a number of technical things. The point is that whenever we think about GOE, we are usually thinking about oxygen. What's GOE is the increase of oxygen on this planet. Well, it is, but it's also not. It's a lot of other things. It's changing minerals, changing what we subduct, changing electron acceptor, changing the availability of the metal that are used by biology to use electron acceptor, to influence the planet, to use oxygen, to produce oxygen. The number of connecting arrows in this complex geo-bio co-interaction, which is the most well-known and the most studied one. GOE is the most studied huge evolutionary event on this planet. It's still incomprehensible. We still have no idea how many interactions are there. We still have a little idea of the different interactions that are there. Just to show you, So molybdenum has been increasing, iron has been decreasing. Have you ever heard about nitrogenases? How many of you? No, maybe you heard about nitrogen fixation, transforming N2 into ammonia, which can be used by other organisms. So nitrogen fixation is probably the single most important step in the nitrogen cycle. We have a lot of nitrogen in the atmosphere, but the majority of organisms cannot use it. There are some specific group of organisms, they use an enzyme to transform N2, break that triple bond, and then make it into ammonia that we and plants and everybody else can use around to do things. Now, think also that the person that found a way to break that triple bond and make nitrate, Haber-Bosch, the Haber-Bosch reaction, won the Nobel Prize for that. Because it changed completely the way we did agriculture and the way we did bombs at the same time. But it's so important that we think that nitrogen fixation, N2 transformation into ammonia, must be a very, very old process. Otherwise, where are we getting the nitrogen from? So if you look into it, these are very complex enzymes, multimeric, lots of protein. At their center, though, they use a catalytic metal center, like I told you, to break the nitrogen bond and then to process it and make it into ammonia, reduce N2 into ammonia and make it bioavailable. There are fundamentally three types of these enzymes. They use different metal. There's a molybdenum-containing enzyme, there's a vanadium-containing enzyme, and there's an iron-containing enzyme. Now, I just told you that metal changed abundance over time. So if I ask you, okay, we have molybdenum, vanadium, and nitrogen, and iron, what's the oldest one? Well, there was a lot of iron before, very little molybdenum, and then they changed it. So you might be tempted to say, well, because there was a lot of iron, it must be the iron one, the old one. That would be a perfectly valid assumption, right? Things get complex, because if you do phylogeny, there's an iron one, but then there's a molybdenum one closer to the base of the tree compared to all the modern molybdenum one coming out. So either we lost some old iron one we don't see, exactly because we don't know what we lost over time, or things are more complex, or enzyme changed availability, or there's another way for molybdenum to be available early on that we didn't think about. The point of this is showing you that even when we start to see the connection, and we think in very linear terms how things are related to each other, things do not always line up so easily. And it's actually a fairly complex interaction, fairly complex story. If you are interested on some of these relationships, there's a paper I wrote not long ago that talks specifically about trace element and biogeochemistry. So GOE is the biggest single most studied coevolutionary event out there, and yet we don't even see well the interconnection between things, because it didn't only change the oxygen on the planet, changed a number of other things cascading down. Now there are other things that have been happening over time. There are coevolutionary events that you might never heard before, because I'm sure everybody heard of GOE, that are as important or more important for some things. Let's think, for example, about, I told you earlier about subsurface life. Let's think about its influence with tectonic and climate. So this is closer to the work we do as a group. This is an image from Costa Rica. Panama is up here, Nicaragua is up here, Panama is down here. And these dots you see here are hot spring we've been sampling in Costa Rica. Now there are a couple of features you should see here. One is the trench, which is the onset of subduction. It's where the oceanic plate is falling under the continent and is falling deep down. It creates this pinch-like structure deep, which is called the oceanic trench. And then once the plate gets deep enough, there are a series of reactions and water is released and volatiles and things like that creates a volcanic chain, which is called an arc. Here we go. Here in yellow are the names of the different volcanoes, and this is the volcanic arc out there. Now, this is fairly common. You might have heard about the Ring of Fire in the Pacific. All the volcanoes, they go from New Zealand up to Japan, Alaska, they go all the way around and back down to Chile. Well, they're all volcanic arc. Those are all oceanic plates that are subducting under the continents and are creating the arcs. We have them in the Mediterranean Sea, Sicily, Greece. We have them in many places globally. They're fairly common. Okay? So we went through this volcanic arc, and we sample springs all over the place, closer to the trench, closer to the volcano in a number of places. Common wisdom has been for a long time that if you go to the top of the volcanoes and you measure the CO2 coming out of the volcano, and you measure the isotopes of that CO2, so how much ratio of 13 to 12 is present on the CO2 of the volcano, you can assume that the CO2 is coming either from the slab that is going down or the mantle. And the quantity is going to tell you how much is being recycled from subduction to the surface and how much is going deep into the mantle. So if you want to make a budget, like a good accountant, you do how much comes out minus how much goes in, and you know how much is being sequestered away. Right? Very straightforward. Now, to me, it was a strange way of doing it because we know CO2 doesn't only come out of the volcanoes, but comes out in every springs. There's bubbling springs all over the place in volcanic area, not just at the center of volcanoes. And when I think about the crust, I told you earlier that we have bacteria and archaea in the crust, not just at the surface, but 5, 10, 20, 30 kilometers in the crust. And this idea of something fractured, porous, filled with microbes, from where microbes are coming down from the bottom, sounds like a lot like an aquarium biofilter, where you have porous media with your bacteria, and you're cycling water through, and microbes are doing things to that water and they're changing it. So we decided to start to track some of these things here. And right now you are seeing the delta 13 of the carbon, so the ratio between carbon 12 to carbon 13 against the standard, against the distance from the trench, which is just measuring the distance of every point, from this line here. So closer to the line, you are close to the trench, up to the volcanoes back there. And then we measured the delta 13 of different things. CO2 gas in the volcano, dissolved in organic carbon, dissolved organic carbon, and all the carbon in the sediments out there. So imagine a pool of bubbling things outside here. We sampled the sediment, we sampled the fluids, we sampled the gases, we just measured everything in a single place. Interestingly, everything we were measuring in the sediment did not change, changing the distance from the volcanoes and the trench, was flat. And if you know something about isotopes, it tells you also that at minus 25 to minus 30, you're measuring plants and dead material from the rainforest in Costa Rica, which is pretty thick. I don't know if you've ever been to Central America. It's as dense as you can imagine a rainforest to be. But if you measure the carbon in the fluids that are coming up from depth, that are coming up in the hydrothermal system, well, the CO2 in the fluids has a relationship with the distance. And the organic carbon in the fluids has also a relationship with the distance. And so interestingly, if you plot the concentration, you can see the bubble size is the concentration against the isotopes of just the inorganic carbon, CO2 in the water, in this case, and the gas, you get something like this. It's a pretty neat line going down, right? It's not a random distribution of things. The beauty of this is that it's spatially segregated. Here are the volcanic gases, and then you're exactly moving from the volcanoes to the trench. So suggest there's a process acting on the carbon as you move from where the plates are colliding all the way to where the volcano is recycling things. Not only, these are two lines, almost parallel to each other. One is further north in Costa Rica, and one is further south in Costa Rica. And we can distinguish them just by looking at the relationship of carbon measured at the surface. Now, for the chemist here, you know rhyolite fractionation, right? How many chemists know a rhyolite fractionation? Okay, we had more chemists before. So basically what this is happening is that heavy isotopes are being removed from the solution, preferentially the light isotopes. So you are reducing the total concentration, but you're also changing the isotopic fractionation of the thing at the same time. And you can model this as being calcite precipitation in this solution, because calcite is precipitating preferentially heavy isotopes. So you could model it, we could do calculation at what temperature fits, what the starting composition of the CO2, and that's a big difference, 5 per mil. And then we did all this calculation, and what you can do, you can say, oh wow, that's a lot of CO2 disappearing here. Because to explain this, you do the calculation, you say about 20% of the carbon we believe was going down is actually stuck in the crust. Now this is interesting, because If you're not going into the atmosphere, you're not going into the mantle, you are in the crust, you are available for microbes to do things. Now, this will make more sense for planetary things as we go across, okay? Now think we were off with our budget. We believed it was going into the deep subsurface for billions of years. It's not. It's staying in the crust, 20% more in calcite. Then I'm a microbiologist, I do extremophiles, and so we looked at the microbes. Have you ever seen a graph like this? It's called multidimensional scaling. How many of you have ever seen a multidimensional scaling? 2 people, okay. Imagine that you wanna look for similarities between me and some of you, okay? You can decide to use one parameter to discriminate between us, for example, height or weight. But the difference between me and you is not only one variable, it's probably multiple variables. Age, height, weight, background, language, you mentioned it, right? If you want to visualize all these parameters in a single graph, you have to make a mathematical construct that create artificial vector of all these parameters together, collapsing them, and then you can plot them on a space. In this space, you can use an index to say how similar are the two of us. Now, if we share a lot of similarities in this space, we are 2 dots close to each other. If we don't share similarities, we are further away from each other, and the further away you are in this graph, the more different you are. Now, this is exactly this exercise, multivariate ordination is called, but done with the number of species, microbial species that are present. A simple way to imagine this is how many species 2 ecosystems share divided by the total number of species. If the total number of the species are all the species they share, well, it's going to be 1, the relationship, they're on top of each other on this graph. If they don't share any species, then it becomes 0, the furthest away possible from each other. So what you are seeing here is a graph that shows you how similar communities in these springs are. The closer, the more similar. And now I just colored it by temperature. Why? Because temperature in biology is important. And we can see the hottest places are closer to each other than colder places, which makes sense. If you are hotter, you share species that like to stay in hot places. No surprises here in biology. Every biologist will tell you that temperature, pH, salinity are major driver in controlling who's present in certain location. And this change in temperature is actually matching the movement from here to here, from the trench to the arc. Because as you get closer to volcanoes, you get hotter things being recycled to the surface. So there's a trend that is explained by the direct subduction. The interesting part is that this was not the only trend we found. We found a number of other trends. And to make a long story short, when we looked at DIC versus DOC here, you could see that if we do pairwise difference between the two, the average distance between the two is pretty constant over time, and is actually always around -9. And isotope people love to measure isotopic signature of different metabolism. I told you the plants have -25. Well, the -9 to -12 is the reverse TCA cycle that Joseph spoke with you about. That's a specific carbon fixation pathway because it's very abundant in subsurface organisms, in anaerobic organisms. So it appears biology is using a lot of the CO2 coming out, even with the temperature pH trend. And if you dig deeper into the single genomes, this one of these line is one genome we reconstructed, and you do calculation of how they distributed only in the forearc, only in the arc, and so on. Turns out you can do calculation about how much CO2 the microbes are using. So then you do simple things. You say, okay, this is the trench going down. This is the 150 isotherm we use, not 122, we use 150. And then power decay law, concentration of cell, depth. You do all your calculation, and you can calculate exactly how much biomass is there, how much CO2, the biomass, the biology is removing, and the total. It turns out that if you do calcite plus the total, it's 40%. Now, why am I telling you all this that it becomes very technical in biology? Well, because a number of you are astrophysicists here and are thinking about, oh, soon or right now, we are measuring atmospheric composition of planet. And that can tell us about biology on the planet. But many of you might incorrectly assume that to detect biology affecting the atmosphere, we need biology to be on the surface. Because otherwise, how is it interacting with the atmosphere? Well, in this work here, we just showed that life that is deep in the crust 5, 10, 20 kilometers, can alter the budget of how much carbon is recycled to the atmosphere, can alter the budget of the methane that is recycled to the atmosphere. So we are not restricted anymore to surface life. We're not. We don't need to think about habitable zone on the surface. There's a lot more that we can do with that. So just by thinking about interaction of life with the solid planet, we can think about exactly, are our assumptions correct? Do we need to look for photosynthesis and surface of planets? Well, probably not. So our space of targets just increased by a lot, which is not maybe a good thing, but it brings diversity. And think about, we've done that measurement only in that place. And if you repeat this, all the red one here are subduction zones, all the dots are volcanoes. If you repeat our work in every other subduction out there, by how much are we off in our calculation? You know, every business survives if it can do a very good job in counting how much money are coming in, how much money is spending somewhere. If there's 40% disappearing in the company, you're not going to last very long. So how are we doing our carbon budget right now might really matter. And not only, think about overtime. Now, these lines here, in the last billion years, are the length of subduction zones in blue over time compared to today average dashed. And this one is ocean ridges, and that's total cumulative crust. And you can see that we had a lot more subduction and a lot less subduction. And if our calculation tells us that the majority of the carbon is stored in this area here, which is called forearc, in front of the volcanic arc, but the total global length of subduction zone changed over time a lot. This means the microbe, in some period of planetary evolution, removed a lot of carbon in the crust. And then in some period of planetary evolution, a lot of carbon went straight to the mantle. What does it mean this? Has been accounted for? Well, for example, a huge change here is related to one of the big change in oxygen. We just said that you need to remove the candy for oxygen to accumulate. So what microbes are doing in the crust with carbon and volatile might also be related on how the planet changed over time. And these are things we are missing to take into account when we build simplistic model on how to look for life out there. Third one is, another example is an interesting one, which is related to sulfur, oxygen, and hyperacidic environment. You know hyperacidic environment, you have heard about them. These are environments on this planet that have a pH below 3, okay? Now, there's plenty of environments like that right now out there. I told you earlier that in natural ecosystem we have pH that goes from 0, sometimes even negative, all the way to 12.5. That's the range of natural pH without our chemistry, natural pH in the environment. Now, you can do very acidic pH in only two ways. First one is magmatic degassing. You have a magma body somewhere in the crust that is releasing acid directly. Hydrochloric, hydrofluoric, sulfuric, bromidric, all the acid you can imagine. They're degassed by magmatic bodies, and that's why we have volcanic hyperacidic lake. The lakes at the top of volcanoes usually are 0 to -1, -2. Super acid, super negative. The other way in which you can actually have acidic places is through biology, the oxidation of sulfide minerals. So you heard about acid mine drainage or any environmental impact due to mining that sometimes get released into the environment, and these water that are hyperacidic and filled with metal, they kill everything. There was a spill in Brazil a few years ago that killed hundreds of people. And you might know of Rio Tinto in Spain. That is hyperacidic because of the sulfide oxidation happening. Turns out sulfide oxidation, if you want to do it abiotically, is fairly slow. So for example, iron sulfide pyrite with oxygen present and water comes iron 2 and sulfate plus H+, this is sulfuric acid, is a fairly slow reaction happening in chemistry. But if you put biology, it goes faster, 10,000, 100,000 to 1,000,000 times faster. It gets really, really fast. And we use these to extract copper, as I mentioned before. Acidophiles, which are organisms, extremophiles that are adapted to low acidity, are well known. They usually like to stay at pH 3 or less. There are both bacteria and archaea there are acidophile, so it's widely distributed in the tree of life, and they appear to be what we call a polyphyletic group. Polyphyletic means they don't share a common ancestor. Okay? Their traits, the group of acidophile is not a single group with one ancestor, it's probably evolved multiple times during the evolution of life. Now, an interesting observation is that the majority of these guys, appear acidophile, is actually using metal sulfide and oxygen to do their reaction. So a group of colleagues in Montana State said, well, this is interesting because if you can only make acid environment with magma or with sulfide biological oxidation, but to do sulfide biological oxidation, you need oxygen, perhaps this is a process that is relatively recent. So they look at the distribution of acidophiles in terms of pH and temperatures in different isolates from Yellowstone, 541 genomes obtained from Yellowstone, and they look at the percentage of how many of the aerobic one are acidophile. And it turns out that the acidophile are in optimal pH, the acidophile are up here, the black dots are genomes that are aerobic, the white dot, the transparent dot, the white dot are genomes that are anaerobic. Turns out that as you drop in pH, everybody wants oxygen. So they put a number of lines of evidence together, and they concluded that acidophile, and we know they do niche construction, because if you grow them in the lab, the pH of your media you grow in them goes quickly down because they're actively producing sulfuric acid. It's a process that emerged together with aerobic respiration. So it's something that happened after oxygen accumulated on this planet. But not only, you needed enough oxygen to sustain these reactions, and probably could not happen before 0.8 billion years ago. So having acidic environment out there, away from volcanoes, is a biological effect. Biology did not adapt to acidic place. Biology created its own acidic place, far away from volcano. Now, why acidity is interesting? Well, it's a great way to break down rocks. And breaking down rocks is a great way to increase weathering. Increasing weathering is a great way to absorb CO2 by silicate weathering, which is a great way to further have an effect on the climate and control the temperature. So you see where I'm going with this, which is very similar to the interaction with climate that we were discussing before. By microbes removing CO2 in the crust and altering how much CO2 is going to the mantle versus how much CO2 is available on the atmosphere. So the evolution of acidophile is another example of coevolutionary event. We discovered it maybe, well, it's been proposed maybe in the last 7, 8 years. There's still a lot of work to be done, but it's a coevolutionary event we didn't thought about before. We just assume acidophile adapt to acidic environment. We never asked ourselves, why do we have acidic environment and why they're distributed this way? And there was a role in biology in creating their own acidic environment. In other words, niche construction and ecological inheritance in acidophiles. I told you oxygenic photosynthesis, the evolution, leading into GOE as a possible geo-bio-coevolutionary event. And that's apparent, and the more you read about it from every different angle, you see there are more connection. We just said the coevolution of acidophile with hyperacidic environment is a coevolutionary event. I will be curious to see if you can come up with any other coevolutionary events. Think about the evolution of some metabolism or something out there that might have had an impact on the environment that then in turn changed the way things evolved after that. I mean, this is a thought exercise, right? We just mentioned evolution of many metabolisms before. How many of these you think were coevolutionary events? How many of these impacted the planet, and then the way they impacted the planet changed life after that? It's not a trivial exercise, because if you go into the literature, there's not much about many of these events. The way we are interpreting right now this event is fundamentally new. Evolution of methanogenesis, when it started? Well, you heard about I'm sure you heard about the faint young sun paradox early on during the evolution of life. Well, if methane was produced by biology early on, that changed our planetary temperature, which for sure changed how biology evolved after that. Sulfate reduction in marine sediments. If you start to subdue sulfur, there is no sulfate, but it's pyrite. You change the redox of the magma, which changes the way you recycle volatile, which changes climate in many ways. Rhizosphere evolution. Without plants and the microbes breaking down rocks, you have less silicate weathering, which means you have less control on the CO2 buffer of the atmosphere. So the list gets going and going and going as you start to think about things, not in isolation, evolution of metabolism, or one specific process happening on this planet, silicate weathering, volcanism, metal availability, but they're deeply intertwined. And there's another one. Sometimes we say that perhaps the Anthropocene is just the results of the evolution of human consciousness. Our ability to influence the environment is a coevolutionary event. We are altering the ecosystem, and the way we alter the ecosystem is changing the way life needs to adapt, and we adapt to it. So it's a two-way street. But it's not the only one. It's been happening before many, many times. So probably the Anthropocene is a new evolutionary event. There's a number of questions remaining. What will be the coevolutionary impact of the Anthropocene on Earth system? To what extent it will affect society? To what extent will impact basic Earth function? These are different questions to me that, especially these two, are two separate set of questions. Sometimes we think we confound them together and we want to answer just by pinning man against environment when we are actually part of the environment, and we should make self-reflecting question more. The bottom line of the co-evolutionary thing is that you should think about the Earth system as a complex set of interacting systems. Not just right now, but also over time, and influencing each other. And if you are approaching looking for life somewhere else, you should think in the exact same way. Impact that are well beyond the single reaction you are thinking about, or the single biomarker you want to use. Because we need, as a scientific community, Earth system science perspective to be able to approach everything we're doing and the challenges we have. Now, the thing we said earlier this morning and the thing we said now, they have a profound impact in the way we approach the search for life in the universe. Huge. And I thought, this being an astrobiological school, and with you having a very different background, but all of you thinking about your future in research, it would have been a good thing for me and you to look together some of the things I told you, how they change or they could influence the way you search for life and we search for life in the universe. So the first one, to me, is that if you take everything we know about this planet's life and its evolution, if life is out there, it's probably microbial. I mean, it's been unicellular for 3.3 billion years on this planet, and it's probably been small for a lot longer than that, even if multicellular. So the probability that life is big and multicellular are not that big. Probably is microbial. So microbiologists, you're welcome. That's a great news for you. Second one is that life is possible in the subsurface, a lot. Now, this is an interesting diagram. Oh, the reference, we're not full screen, so you cannot see the reference. I have the reference down here. This is a work by McMahon and Parnell, where they looked for distance from the star in astronomical unit and temperatures, right? When we think about habitable zone, usually we think along this line up here. How far can we go from the star to be in an area where we have temperature condition on the surface that allow for liquid water to exist? Now, if you assume that you have liquid water also in the surface, and it's not anymore an assumption since we found it on Mars, and we know it's on Enceladus and Europa, and probably a number of other places. For planets that are 10 times our own mass, identical to Earth and 10 times less, how far away we can go from the star and still have subsurface temperature at kilometers depth that are between 0 and 122. And so you're not restricted to a very small set, because even if you don't go that deep, only 1 or 2 kilometers, you are moving. You are moving by 1, 2, 3, 4, 5, 6, 10 astronomical units away from the star. Now, I agree with cutting away the possibility of oxygenic photosynthesis of being on the surface as you move too far away from the star. But at the same time, we are showing that there could be a lot of life underground, recycling things that are produced by geology. So if a planet is geologically active, there might be water underground, and it might be very far away from a star, or might be completely frozen on the surface, and yet life might theoretically still exist. So thinking about subsurface life should help you to change your definition of habitable zone and expand it a lot. This is well known and studied. And as I told you, we found water underground on Mars, so we know this is not just theoretical even on a rocky, dry planet like Mars. So this is our classic habitable zone, and probably in reality we're gonna need to broaden it a lot more than we do normally. Now the other thing is that when we say what life needs, usually we say you need water, you need building blocks, the so-called SNOPs element, carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur, and you need thermodynamic disequilibria, right? This is the mantra for where to look for life. I think the last one is the most interesting one, because if you need thermodynamic disequilibria, you need to access it. And we said that all life that we know interacts with thermodynamic disequilibria in the term of redox chemistry. So we are looking for redox, essentially. That's what we do when we think about hydrothermal vents inside Enceladus, doing water-rock interaction or atmospheric composition somewhere. But life, in order to interact with redox, needs metal, especially trans-iron metal. And all the astrophysicists here know that trans-iron metal, they're not made in normal star in good quantities. You need big events. You need supernova. You need merging neutron star. And so with a colleague, Dario is doing his PhD with him, Giovannelli, we started looking for the distribution of nickel, for example, against total metallicity of stars in stars catalog, and looking for the distribution in rocky planets. And things are not linear. There are some interesting patterns there. And perhaps thinking about the necessity of life, of having metal to control redox, could be a further step that we add, a further filter that we add when selecting target to look for life. Needs to be a place where there's enough trans-iron metal that planetary processes don't sequester all of them in the nuclei and leave some on the surface for life to control redox. Otherwise, you cannot control redox. So there's a preprint out there with this one, if you want to read it. So water, building block energy, metal availability. And these are two examples looking at nickel against total metallicity and nickel against carbon in the same systems. The good news for the people that have been obsessed about detection of atmosphere is that even if life is subsurface, if you have an active geology, that's the caveat, we're going to still be able to detect it. Now, you know plate tectonics here. I told you plates go under other plates. They create volcanoes. Volcanoes emit gas. They influence the atmosphere, and there's a cycle. This will be our modern plate tectonic. This is not the only tectonic possible. There's a number of other tectonics that are probably out there that are important and have been important. The bottom line is that if you are recycling the crust one way or another, you are recycling volatile from inside to outside, then even if life is in the crust, you're going to see the effect in the atmosphere. Now, the only problem is this one, terminal stagnant lead tectonic, where if you are a geologically dead planet, if you don't have recycle of material at the planetary scale, there's no communication between deep and the top. But this means life is on a one way to death, because there's no way to recycle element and redox gradient. Once they're gone forever. The way in which we're doing things here, we are subducting things and reassuming them that are reduced, subducting oxidants and resuming reductant. That's why our planet keeps going over time. But as long as there's active geology, we're going to be able to detect life, even if it's deep. And we already did. We already did detect subsurface from surface. Think about Enceladus. We did flyby in the south, and we detected composition of the plumes, and we were able to make inference about things happening under the ice at the interface between water and rocks. That's because there's an interface of activity that recycle things from inside and out. And so as long as that's true, we're going to be able to detect imbalance in atmospheric chemistry, even if life is not on the surface. So no need to obsess with oxygenic photosynthesis and surface life. Let's keep our mind open. And even Mars, we know there are Marsquake. Small, but there's something still moving. And a lot of my colleagues bet that if we ever gonna find life or sign of life on Mars, it's gonna be in the subsurface. And now there are proposals to go drill between 2 and 20 meters depth to be able to get below the zone where radiation is destroying everything and every possible evidence. The other thing is very practical. If you think about the way microbes interact with the periodic table and the diversity of reaction they can do, it means they're going to be very important for space colonization. You heard about in situ resource utilization as a concept, probably. How many of you have? Not many. Okay. The idea in space colonization is that you're not going to be able to bring everything from Earth. It's heavy. It's difficult to bring. It's limited. You need to access the resources that are on the planet. The easiest one to think about is water. Melt the ice and have the water, and then split the water, you get the hydrogen and the oxygen to breathe, and so on. But these are not the only thing. We're going to require mineral. We're going to require mining. We're going to require to make soils. We're going to require to do a number of things. And we know microbes are great at doing things fast. So we can think about doing biomining in space to extract minerals that are necessary for 3D printing. We're going to need to convert regolith into soil that are not toxic for planting and plants. We're going to need to produce food and oxygen one way or another. And all these, which technically is called in situ resource utilization, goes through microbes in this case, and their ability to interact, which is not limited to biologists to study, but we're going to need engineers, we're going to need chemists, we're going to need physicists, we're going to need everybody to study on this. Because there's a problem with what light we're going to have available. There's a problem of what catalyst we're going to need, what material we might be able to make. There's a lot to do. It requires, again, an integrated system. And then finally, one of the greatest things that comes out of thinking about life as being a coevolutionary event, and this goes back to a nice question earlier on this morning, is that we think that life enables life to exist. So you know the Drake equation, right? The number of planets that have life out there, and you set all these parameters, and then yet, for how small you put them, we should see a lot, and we don't see anything. So for a long time, we've seen this as being an emergence bottleneck. So this is the fraction of planets that are within habitable zones. This is time from start of the planet to 9 billion years. And then we are saying that most planets that are habitable within habitable zones, over time, they never had life. Because life is very difficult to start. It's very unique. That's the uniqueness of life. So we have a very small fraction of planets that ever had life, and then it goes extinct at a certain point on. This is called emergence bottleneck. This is the assumption we used so far in discussing life in the universe. Life is difficult to start. There are alternative hypotheses. One hypothesis is that there's no emergence bottleneck. Life is easy because life is a planetary response, right? Planets need life to dissipate things. So if you do this, very early, like Earth, many planets, a high percentage, 80% or something, we can be arbitrary on that, they must have life on them, and then it's going to get lost later on. So life should be very common in the universe, but well, we haven't seen it yet, right? It might be common, we haven't seen it yet. So this is emergence bottleneck. This is no bottleneck. Life is easy. But there's a third option, which they called, the author of this paper, Chopra and Lineweaver, in their paper, they discuss this very, very well. They say there's a third option here, which they call the Gaia bottleneck. The idea is that, yeah, life is easy to start. It's a planetary necessity. Starts everywhere very quickly. The problem is that life and planet don't really play well most of the time. And so life gets extinct very, very quickly. Like Mars and Venus, they go into runaway scenario. Boil their ocean, lose their water, freeze completely. Things happen for which then life gets extinct. So we don't see life in a lot of places, not because it never emerged there, but because the interaction between life and planet never stabilized. And we've made the example of temperature on this planet. And we've been frozen many times as noble Earth, and yet came back. That's a clear sign of biology helping to regulate the planet in one way. That's why they call it the Gaia bottleneck, after the Gaia theory. Now, this will be that the conditions that are habitable and the planetary environment, they stay together, and life emerges, and then things go lost, early extinction of Gaian bottleneck, but sometimes life and planet keep working together, and somehow the planet stays in the trajectory just long enough for life to reproduce and evolve and become more metabolically complex, if you want. I'm not talking about the biodiversity out here, talking about biodiversity of metabolism, which can stabilize the planet. Now, if we assume this is a possible option, what we need to look for is not sign of life, period. It's sign of planetary interaction with life. It's sign of regulation of some of the things. If a planet should be frozen and is not, and our reaction network don't show that geology alone can sustain those rates of methane being present, is much more powerful than just thinking about methane and oxygen being together at the same time. It's more complex because you need a deeper understanding of Earth system science and how they interact and chemical reaction network and parameters. But thinking of life as something that is easy to emerge, but the life-planet interaction is what controls its longevity, is an interesting way to approach this. And it's a way that is enabled by thinking about life as a coevolutionary event. Now, the idea of coevolution is a new one in science right now in the term of how we are exploring it, but it's not new in science. As for Thomas Gold discussing the deep subsurface biosphere, not being a biologist, the theory of life and planet coevolution, in a way, is born through a Russian geochemist, Vladimir Vernadsky, which in 1926 took a term, biosphere, which was coined by Suez, an Austrian geologist, to mean the thin layer of biology on top of the planet, and brought it to another level. Talking about the biosphere as the layer of the planet where biology and geology interact in a way in which you cannot separate the two from one another. It's a very interesting book. It was translated only in the 50s in English, so it was unknown for most of Vernadsky's life. It was translated by Vernadsky's son. I think it was very influential in creating the Gaia theory and a number of other cybernetic and well-connected concepts about planetary system. It's a short read, but it's an intense one. And I suggest you read it. Because what Vernadsky says, and this is me paraphrasing the entire book, is that life is a geological force that has been modifying our planet and still shapes our planet. So in this concept of coevolution, life is not something that happens to the planet, is intimate and intricate part of the planet, and is actually one of the main geological forces that changes the planet. Now, to tell you how much this is profound, I was having a conversation with a colleague, Shona Morrison. She's a mineralogist in the US. She's well known in the community. She works on Mars. She's responsible of the Chemin instrument on the Mars rover. She has worked on mineral evolution with Robert Hazen, and they published a number of great books. And we were discussing the possibility of using some place on this planet as mineral network that are completely abiotic. In other words, they don't show in one parameter, be it their isotope, composition, or whatever trace element, sign of life. And in our discussion, we realized that probably right now, there's not a single place on this planet that is abiotic in signature. I mean, we found diamonds that are 3.5 billion years ago. They come from ultra deep inside the mantle, and inside we find the isotope that are suggestive of life and life material being subducted. So life has been influencing so much of this planet that not even fishing deep into the mantle, we're going to be able to see purely a biotic signal of the planet that was before life, because we've been subducting carbon material and life material for a very long time. So life is a geological force that is still shaping our planet. And that will be it, and all the questions you want.